Welcome to this explainer. Okay, today we are diving into a 3.5 billion-year-old mystery that literally powers our entire world. We're going to unpack the absolute marvel that is photosynthesis, and we're going on a trip from the massive global scale of the Earth's carbon engine all the way down to the mind-bending quantum secrets hiding inside a plant's microscopic cells. Let's get into it.
Okay, let's dive into this. Here's our roadmap for today. First, the global carbon engine. Second, a timeline of discovery. Third, the photosynthesis process itself. Fourth, C3, C4, and CAM plants. And finally, the quantum secrets of photosynthesis.
All right, part one, the global carbon engine.
Before we get bogged down in the microscopic molecules, we really need to look at the Earth as a whole interconnected system. Just take a look at a lush wetland environment like this. It perfectly illustrates what scientists call the fast carbon cycle. Plants and tiny marine phytoplankton, they act as the main biological engines here. They're constantly absorbing carbon dioxide and sunlight to create fuel. At this macro level, it's a beautifully simple equation. Carbon dioxide plus water plus solar energy equals sugar and oxygen. Then when those plants decay or burn or just get eaten by something, the exact reverse happens. Oxygen combines with that sugar to release water, carbon dioxide, and energy right back into the system. But what's absolutely amazing here is that this cycle is so tightly bound to plant life that our atmospheric carbon dioxide literally fluctuates with the seasons. I mean, in the spring, when plants are growing like crazy and pulling in carbon, global CO2 levels actually drop. And in the winter, when leaves die and decay, those concentrations climb right back up. It is literally like the Earth itself is breathing.
So you might be wondering, how exactly does a process this ancient end up powering our entire modern world? Well, research by Dr. Tanai Cardona at Imperial College London reveals that the earliest form of this mechanism, something called anoxygenic photosynthesis, which used molecules like hydrogen sulfide instead of water, actually evolved more than 3.5 billion years ago. Think about that for a second. That is before most groups of modern bacteria even branched off. It tells us that during the entire first half of Earth's history, the vast majority of life forms were probably capable of some form of photosynthesis. It basically laid the ultimate biological foundation for almost all life today. But since humanity wasn't exactly around billions of years ago to watch this happen, how did we actually figure it all out?
Let's look at part two, a timeline of discovery. It took centuries for scientists to really piece this massive puzzle together. Way back in 1648, a guy named Jan Baptista von Helmont finally figured out that plants grow by accumulating water, not just by eating dirt like some sort of really slow green vacuum cleaner. Fast forward to 1771, and Joseph Priestley discovers that plants release oxygen. Or, as he put it back then, they restored combusted air so it could be breathed again. But the real breakthroughs in the actual chemical mechanisms, those came much later. In 1931, C.B. Van Niel proposed that the oxygen released actually comes from the splitting of water, not from carbon dioxide like people thought. And then, by the 1950s, using radioactive isotopes, Melvin Calvin fully mapped out the path of carbon fixation, the very process we now know as the Kelvin cycle.
Let's move to part three and see how this builds. The photosynthesis process.
Okay, let's zoom way down into the microscopic machinery of the chloroplast to really understand this. Essentially, the whole process of photosynthesis is split into two perfectly complementary phases. Step one is the light-dependent reactions. This is where light energy is trapped by chlorophyll in excited, high-energy electrons. This creates biological energy currencies called ATP and NADPH. Then, step two is the light-independent reactions, often called the dark reactions or the Calvin cycle. This is where the plant takes that freshly minted ATP and NADPH and uses them to actually fix carbon dioxide into complex carbohydrates, like glucose.
And the absolute star of that Calvin cycle is an enzyme with a remarkably long name, ribulose 1,5-bisphosphate carboxylase oxygenase. Yeah, let's just call it Rubisco for short. Rubisco is the critical enzyme that actually grabs the carbon dioxide coming in through the leaf's pores and remixes its components into organic molecules. It's so essential to the global carbon engine that Rubisco is actually thought to be the single most common protein on planet Earth, literally everywhere.
But here's the catch. Before Rubisco can do its job, the plant needs that initial spark of energy. And the visible color of a leaf gives us a massive clue as to how it gathers it. If we look at a plant's absorption spectrum, we can see exactly how it catches solar energy. Pigments inside the chloroplast, specifically chlorophyll A, chlorophyll B, and carotenoids, absorb tiny packets of light energy called photons. But they don't just absorb all light equally. They mostly soak up the blue and red wavelengths while bouncing the green ones right back out. And that right there is exactly why healthy plants appear green to our eyes.
Now, what's really interesting about this next part is how that absorbed light gets utilized in what's known as the Z-scheme. Down here in a protein complex called photosystem 2, a photon of light hits a reaction center and excites an electron to a much higher energy state. Now, because that energized electron shoots off to travel down an electron transport chain, the plant has to replace it. And it does this brilliantly. It literally splits a water molecule using an oxygen-evolving complex, rips its electrons away, and releases oxygen as a byproduct. That is the exact oxygen that you and I are breathing right now. Pretty incredible, right?
So as that electron travels along the chain, it helps pump protons to create ATP. But its journey is not over yet. It eventually arrives at a second complex, appropriately named photosystem 1. Here, it gets hit by another photon of light and gets excited all over again. This second massive boost of energy is ultimately used to reduce a molecule into NADPH. So together, that ATP and NADPH are the powerful biochemical batteries shipped right over to our old friend Rubisco to power the Calvin cycle.
All right, let's look at part 4, C3, C4, and CAM plants.
Now, as incredible as this biochemical machinery is, it's actually not completely flawless. There is a massive biological glitch in standard photosynthesis, and it all comes back to Rubisco. See, if carbon dioxide levels drop inside the leaf, like when a plant has to close its pores to conserve water on a really hot day, Rubisco gets starved for CO2. And in these conditions, it gets confused. It'll just happily bind with oxygen instead. This process is called photorespiration, and it is a huge, wasteful mistake for the plant. It produces 25% less product, burns up valuable ATP energy, and even creates toxic waste like hydrogen peroxide. The scary part? Most plants in the world, including crucial food crops like wheat and rice, are standard C3 plants that suffer from this exact flaw. So as global temperatures rise, these plants are forced to close their pores more often, which triggers this wasteful photorespiration and severely threatens our global food supply.
But nature is remarkably resilient. Certain plants, known as C4 and CAM plants, actually developed incredible evolutionary workarounds to avoid this whole photorespiration mess. They do this by using a totally different enzyme called PEP carboxylase, which isn't tempted by oxygen at all. C4 plants, like corn and sugarcane, thrive in hot climates by spatially separating their processes. They use a specialized leaf structure called Cran's anatomy to fix carbon in one cell, and then they pump it into protected bundle sheath cells where the Calvin cycle happens. This literally physically keeps Rubisco safely away from the oxygen. Meanwhile, CAM plants, think desert succulents or pineapples, they separate the steps by time. They open their pores at night when it's nice and cool to collect carbon dioxide, store it as an acid, and then use it during the day while their pores are tightly closed to prevent water loss. Pure evolutionary genius.
And finally, part 5, quantum secrets of photosynthesis. All right, we are pivoting to the bleeding edge of physics here because it turns out that how plants transfer solar energy into those reaction centers isn't just basic chemistry. It actually involves quantum mechanical effects. Because of this, green plants and cyanobacteria can transfer that sunlight energy through their molecular systems with nearly 100% efficiency. That is just completely staggering. The transfer happens almost instantaneously, meaning practically zero energy is lost as wasted heat along the way. To put that into perspective, it's a level of energy efficiency that our best human engineering hasn't even come close to matching yet.
Which is exactly why scientists are so obsessed with this right now. As researcher Gregory Engel noted, nature has had about 2.7 billion years to perfect photosynthesis. This gives us a new way to think about the design of future artificial photosynthesis systems. By studying the intricate, mind-bending quantum mechanics of this ancient biological process, modern scientists are actively drawing inspiration to design future, highly efficient clean energy systems for our own species.
So this all leaves us with a massive, world-changing question. Can humanity unlock these quantum secrets to build perfect artificial photosynthetic systems? Think about it. If we can harness and replicate the exact same process that fueled the Earth's global carbon engine for the last 3.5 billion years, we just might be able to save our own rapidly changing climate. It's a deeply provocative and fascinating thought to leave you with today. Thanks so much for joining me on this explainer, and keep questioning the incredible world around us.